All right. Welcome to our Geriatrics Grand Rounds. A couple announcements before we hop in. Our next Grand Rounds is February 6th, and that will be Noel Javier talking about inclusive and affirming geriatric and palliative care for the LGBTQI plus community. Other announcements, Geriatrics Works in Progress is going to be upstairs in 8200. And after today's grand rounds, you'll receive an email with a link to the recording of today's presentation. Also included in the email will be a link to an online evaluation. Please complete the evaluation if you can. And please submit any questions you have about today's presentation in the chat or the Q&A boxes on Zoom, and we'll get to those, assuming that we have time. So without further ado, I would like to introduce Jonathan Kurche, who is an assistant professor in the Pulmonary Sciences and Critical Care Medicine division at CU, and he is also a 2024 Geriatrics GREC Advanced Research Fellow. His primary clinical appointment is at the VA, where he is in the pulmonary division, and he has an affiliated appointment at CU. He has experience caring for patients in the medical intensive care unit at Rocky Mountain Regional and at CU Hospital, and sees patients with advanced pulmonary disease in the chest clinic at VA. He's currently the VA representative to Panel B on COMERB, and Dr. Kurche's research focuses on the pathogenesis of fibrotic lung disease. His specific goal is to understand how mechanisms of aging in the lung influence development of idiopathic pulmonary fibrosis. Dr. Kurche is mentored by Dr. James Gregory, a cancer biologist and biochemist who studies the role of clonal evolution in hematopoietic and lung cancer development to understand how age-associated, acquired, and somatic mutations impact pulmonary biology. Jonathan is currently supported by the GREC, by CCTSI, and Cancer League of Colorado, and he has no industry conflicts of interest. John trained here for his MD and PhD at CU, and got his undergraduate degree at Kansas State. So without further ado, the topic is acquired mutations in aging and age-associated lung disease. Dr. Kurche.
All right. Thanks, Josh. So I am, by sort of origin, a basic scientist, and I am in an emerging area, I think, of both aging and kind of basic science research. And so what I'm about to present to you is essentially kind of a works in progress talk, but I think I've sort of wrapped it in some clinically relevant information for you guys. But of course, bear with me. This is sort of my disclosure slide. I have to give a shout out to the CCTSI. Much of this work wouldn't be possible without their help. And then David Schwartz and James DeGregory have done a lot of the legwork to produce some of the specimens that we've used in this study. And of course, as a VA investigator, I'm hoping to sort of transition over to more sort of VA-oriented care and focus, but it's really useful to have some of the quality of specimens and quality of patient history that we're able to resource here on this campus. So a lot of this work is contributed by folks who are outside the VA system. So the learning objectives for this talk is I want you guys to be able to understand kind of the difference between different types of genetic variation in the human genome. And I think some of you might be wondering, like, what is the relevance of that to kind of like my clinical practice, to kind of my sort of framework of how I view aging. I'm hoping to make that clear to you as we kind of go through. But I think the overview sort of statement is that there are diseases that we see in aged individuals, some of which have been set up by genetics and just lie dormant throughout most of the health span until, for various reasons of sort of declining quality of the cells that make up the immune system, or environmental exposures that you acquire throughout your lifespan, some of the barriers to making those phenotypes evolve and erupt kind of break down. The other thing I want to say is that all the experiences that we have over the course of our lifespans leave a mark, and some of them leave a mark within our genome and within the cells that make us up. And I'm hoping to sort of help make that clear kind of as I go through my talk today. And so my overall goal is to kind of understand how aging influences lung disease. And when I say lung disease, I'm talking about things like lung cancer. I'm talking about things like idiopathic pulmonary fibrosis. I'm talking about COPD, kind of the big three diseases, really the big two that I manage in clinic, because most of the cancers tend to go towards our oncology colleagues. And the goal here is to try to understand not only why these diseases evolve and why they manifest in predominantly aged individuals, but ideally to be able to leverage that knowledge to improve the health and well-being of our patients. So I'm going to start with a case. So I'm going to tell you about an 84-year-old guy who presents to my clinic with cough and shortness of breath, or he might be presenting to your clinic with cough and shortness of breath. This is not a phenotype that we don't see with some frequency, right? And so this is a guy who's got a relatively benign past medical history, some high blood pressure, some dyslipidemia, some prostatic hypertrophy. Of course, we've done all the things that we need to try to limit how many pharmacotherapies this individual is on. And we've used shared decision-making to make sure that we are, doing what is best for them in terms of their health span. This individual presents with a cough that's been going on for about 2 months. He thinks it may have started with kind of a URI, did some symptomatic relief for that, maybe took some cough suppressants, but it's just been kind of nagging and hanging on. And he's got, a little bit of fatigue that's associated with this. He's noticed that, he can't quite do as much in the yard anymore. He can't quite do as much when he goes to the grocery store. He's leaning over on that grocery cart a little bit more and having a hard time bending over to pick up the groceries. So this guy smoked for about 40 pack years, but heroically was able to quit tobacco about 20 years ago. He normally enjoys gardening. He takes care of his wife, who's got breast cancer and is undergoing treatment for that. He's got a couple of adult children in the area, but he's taking care of all of his independent activities of daily living. These are his meds. And then when you get him kind of up on the exam table, you look over his vitals. The saturations are a little bit low, even by Colorado standards. We'd expect normals to be kind of in the 92 to 93% range. He's at an 89 at rest. Something's going on. But you look him over, he doesn't have any evidence of kind of overt heart failure. He's got some adventitial sounds in his lungs when you listen at the bases. Otherwise, no specific wheezes or anything like that. And then his neurologic exam for somebody his age is essentially benign. So what are you thinking right now? You might be thinking about this guy having a cough that started with an upper respiratory infection. You might be thinking like, okay, is there some chronic rhinosinusitis that I need to take care of here? Is this a guy who probably had long-standing esophageal reflux disease and maybe he's been staying up later and eating more because of stress related to caring for his wife or something along those lines. So he's not doing the esophageal hygiene things we think people should do. He's having snacks right up until bedtime and then he's laying flat and stuff's coming up and he's waking up and he's got an irritated larynx. Or maybe you're thinking like, is this a guy I need to send to speech for a swallow evaluation? You might ask him a little bit more about whether or not he's choking or coughing more when he's eating. You might ask him more about when this cough is actually presenting. Is it in the middle of the night? Is it happening during the day? Is it kind of sporadically throughout the day? You might be thinking about asthma and airway type reactivity diseases. Can asthma present in somebody who's 84 years old? Yes. Can it present in somebody who's recently had a viral infection? Yes. Do the same medications that work for asthmatics tend to work in individuals who are older, who may have had sort of cryptic asthma their entire lives and just got used to not being able to breathe as well as the people next to them? Most of them do. There are some caveats. You want to be careful about folks who have rapid AFib and giving them, an albuterol inhaler, but those are things that can be worked out. You might think about chronic bronchitis. Is this just latency from his COPD manifesting now? Emphysema, bronchiectasis, a disease of the dilation of the airways. It's often caused by immunosuppressed conditions. HIV can be caused by chronic aspiration, can be caused by chronic variable immunodeficiency, Ig deficiencies, things like that, things that can manifest in older individuals. And then you may think about pulmonary fibrosis. You may think, is this a guy who's got a secondary pneumonia? Do I need to get some imaging? Do I need to kind of help figure out sort of what's going on? I also would just prompt you a little bit. Sometimes cardiac arrhythmias can present with coughs. If you're having frequent palpitations, I've met a number of individuals that was the sentinel finding, was this cough, but it was because they were actually going into runs of supraventricular tachycardia. So what are you going to do? You're going to titrate his oxygen. You're going to get some imaging, probably. You might get some pulmonary function testing, depending on what institution you're at. You may or may not be able to order that on your own, and I'm sorry about that. You might consider a barium esophagram. Barium esophagams, I would say, are pretty useful if you're looking at either reflux disease or dysphagia. So think about having the radiologist lay them flat, seeing where that barium goes, because even if they don't have transfer dysphagia, it might be coming back up. You might counsel them about esophageal hygiene. You might prescribe some PRN medications. You might consider talking to a pulmonologist. So you get an X-ray. It looks like this. I stole this from the Cleveland Clinic. And these arrow signs, I think, in this X-ray are pointing to peridiaphragmatic honeycomb cysts and a little bit of fibrotic thickening in the minor fissure of the right middle lobe. But I'll show you that in more detail right here. So this is what your imaging looks like. So this is an 84 individual who has pulmonary fibrosis until proven otherwise. And when we look at CT scans of individuals who are kind of in this age range, we sort of think like, okay, is this somebody who might have pulmonary fibrosis based on some thickening, scarring that we can see kind of within the lungs? Is this somebody who probably has pulmonary fibrosis, which is based on, absence of features like ground glass, the presence of features, like the fibrotic thickening, and kind of an age range that's appropriate for individuals, which I'll touch on in a second? Or is this somebody who definitely has pulmonary fibrosis? And we use the words pulmonary fibrosis and UIP these days interchangeably. So what's UIP? Usual interstitial pneumonia. That's a pathologic diagnosis. You have to look at the tissue under the microscope. Pulmonary fibrosis is much more of a radiologic and clinical diagnosis. But if you've got all the features, honeycomb cysts, absence of ground glass, fibrosis, that is UIP until proven otherwise, usual interstitial pneumonia. When you get a biopsy, most of the biopsies show you that you have usual interstitial pneumonia disease. And the key variable here is that you're looking at somebody who's over the age of 50. And in particular, if they're over the age of 80 and they have features like this on CT scan, you don't get a biopsy. In most of these cases, we don't get biopsies anymore because of work like this. But the point is that it's idiopathic pulmonary fibrosis. You were completely justified in treating their disease that way. Now, why is that important? Well, IPF has, at least a 50 or as high as a 50% 5-year mortality. So there are a couple of therapies that are available. They work okay. We can talk about those towards the end of the talk. But what I really want to focus on actually is this feature of the disease, that the pretest probability of looking at a CT scan like this and calling it pulmonary fibrosis and knowing it's pulmonary fibrosis on a biopsy shoots up at about age 50. And I want to talk about why that might be the case. So idiopathic pulmonary fibrosis is basically a genetic disease. There are a number of alleles that we know that are associated. Over 30% of the cases are heritable. The alleles that don't account for that heritability are, polymorphisms in genes like MUC5B, which David Schwartz on this campus studies, polymorphisms in genes that you guys may be familiar with, like TERT and TERC, the telomerase components that sort of regulate the length of the ends of telomeres in our genome. And so the idea is that most people go through life until their 50s, 60s, not knowing that they carry these polymorphisms. They probably shouldn't know that they carry these polymorphisms. But something happens when they hit their 6th, 7th, 8th decades of life. The epithelium gets injured, usually by a sort of a viral prodrome or an association with a viral prodrome, sometimes by surgery. And that epithelial barrier starts to break down. And rather than healing normally, like our lungs do when we get rhinovirus from our kids, or we get RSV when we're kids, or we get a pneumonia or something like that, they stop healing functionally and they start to form this dysplastic sort of scarring tissue that we see on microscope. And there are certainly things that can sort of amplify this fibrotic response. We think of things as like smoking. There's probably an air pollution component, although that hasn't been completely worked out. And aging almost certainly kind of accelerates this genetic susceptibility into manifesting in this disease, but it doesn't do it all the time. So the genes that we think about that are most sort of prominent in idiopathic pulmonary fibrosis, that are germline genes, meaning you get them from both your parents and they're in you from the second that you start to develop as a zygote, are polymorphisms in things like desmoplakin, which binds cells together, MUC5B, which I mentioned. You can see it's the largest value on this chart. There's a protective allele in the telomerase processing enzyme. And then there is a harmful allele in the template that your cells use to replicate telomeres at the ends of your chromosomes. And then a couple of others on here. This is not the main topic of my talk. I want to talk about non-germline variants, but certainly the germline does set us up for this disease later in life, and it remains an open area of investigation why there's such a long latency time and what it is about our exposures that lead people to develop this disease. So the normal pulmonary epithelium, we tend to think of it as being divided into kind of the alveolar tissue, the air sacs. Let me see if I can bring up my laser pointer here. Ta-da. Okay, and so those are relatively simple. They're composed of two different cell types, lined by an interstitium that has fibroblasts in it and capillaries, of course. And then we think about airways, and airway cells are much more complicated, at least as we understand them now. We have different cell types for different functions in the airway. In the distal alveolar spaces, we essentially just have two cell types, one which is the stem cell that makes a surfactant, the alveolar type 2 cell, and one which is the thin cell that makes up the membrane, the alveolar type 1 cell. But this organization gets really sort of scrambled in individuals who develop this disease. And we start to see mucus secreting cells way down in sort of the alveolar spaces where they shouldn't be. That's part of what gives rise to those big honeycomb cysts that we see, are dysplastic cells along those lines. We see thickening of this fibrotic interstitium, and our alveolar type 1 cells that make up those thin lacy membranes that you guys may remember from pathology classes, those disappear, and it's unclear what kind of drives this process. So taking a step back, idiopathic pulmonary fibrosis certainly manifests in older adults, but it's not the only lung disease that manifests in older adults. So I grabbed this from Trinetics through our Compass system, just looking at hospital admissions here at UC Health. And what you'll notice is that our admissions for chronic obstructive pulmonary disease, our diagnoses of IPF, and our admissions for pneumonia all follow the same pattern. And that is that right about age 50, these things go off the charts. And I think, you know, the implicit biology in this is really interesting. And I sometimes have folks ask me questions like, why are you trying to cure aging? It's a natural part of life. And I don't think that's the impression that I want you guys to take away from this slide. But I think if you're going to think about like how people function through the end of life and how health span sort of evolves over time, this inflection point is probably a place where we want to do a little bit of work to understand what the basic processes are, you know, that are influencing these things. And so, because it really does make up a big proportion of what our healthcare system is here to do. So people have tried to generate frameworks of what it is about aging biology that ultimately dictates the aging process. And I think the global view from my perspective is that it's unclear, to say the least. So people have invoked a number of different kind of basic mechanisms, and this is, of course, from a review last year that was kind of a modification of an earlier review that I think occurred in like 2010 about kind of what the basic biology of aging is. There's no specific gene that drives aging. I'm sure if there were, we'd probably have a pill for it. There is, at least as far as we can understand, but there are features of aging that we all recognize, and we recognize this when we sit across from our parents at the Thanksgiving table. We recognize this, you know, to some extent when we're seeing patients in clinic, and that is that we don't heal as well, the cells get exhausted, things aren't sort of firing the same way in older individuals, and cells aren't communicating the same way that they do in older individuals as they do in younger individuals. There's an inflammatory component to it. Folks are prone to sort of infections, bacterial overgrowth. Things start to break down. And the way that this review sort of encapsulates kind of these hallmarks of aging is that there are some main drivers, and those drivers include things that I've already hinted at: loss of telomeres, those sort of replication ends at the ends of chromosomes, changes to the epigenome, changes basically to the way that our DNA is programmed to be expressed at certain times in cell cycles and developmentally, inability of our cells to clear bad protein-related garbage that sort of builds up over time, particularly in our long-lived cells like neurons. And then essentially chromosome breaks, changes to our genetic code. And all of these things have been things that have been investigated to one extent or another. So when I started approaching pulmonary fibrosis research, particularly from an aging frame, I thought, maybe there's an epigenetic relationship. So we have all of these epigenetic clocks that we can use to measure, how old somebody is molecularly. And it seems to correlate pretty well with how old they are physically. And there are different features of these clocks, and I'll just sort of briefly drive into that. But different investigators have come up with different, essentially clock-like programs to look at methylation patterns across the genome. And you can come up with a score, and you can do a regression. You can figure out what the approximate methylation age of different individuals is. So when we look at lung tissue, and we look at whether or not age correlates with the methylation age in the lung tissue, we find that kind of regardless of which clock we pick, they all do a pretty good job of predicting how old that lung tissue is in a control specimen. But the problem seems to be that if we take one of these idiopathic pulmonary fibrosis specimens and we look, we might predict, because the cell is scarring, it's fibrosed, there's all these telomere changes that we think are associated with IPF, we might predict that the tissue looks older on a methylation basis. And that's not the case. So not all clocks are exactly the same. If we use Steve Horvath's sort of now classic clock from 2011, he was the first person to kind of come up with this idea that you could score these methylation marks. It actually shows that the pulmonary fibrosis lungs look younger from a methylation standpoint in bulk tissue. Now, what does this mean? I'm not here to get into like the details and vagaries of these scores necessarily, but if we use another clock, for example, the frailty clock, it does show that these lungs are appreciably more frail than the lungs of a control subject. So it kind of depends, right? It depends on which clock we choose, how we interpret the data. If I wanted to stand up here and just tell you that the clock predicts that these lungs are actually older, I would just give you the Levine data, and maybe we wouldn't talk about the Horvath data. But I think the point is that methylation-related data in this disease may be a little bit inconsistent. It may not be the gold standard driver of what's going on in pulmonary fibrosis. And so it prompted me to start to look for other potential causes. And so getting back to this, I started to think about genomic instability. And here is sort of overlaid kind of collective epidemiologic data about how old people are when pulmonary fibrosis tends to manifest. And you can see there is a clear age association. And I think the message that I'm hoping you guys will take away from the talk today is that we're all mosaics. So as we go through life, our cells acquire their own mutations, and they carry those forward in the daughter cells that they produce. And those mutations can be consequences of exposures that we have, smoking being one of them, air pollution probably being another, chemotherapy certainly, radiation certainly. But this is an interesting area to study, and it's relatively understudied. And so I thought maybe this is a place where I might find some signals for pulmonary fibrosis and what's prompting this illness. So back to the learning objectives, the difference between somatic and germline variants. Germline variants are variants you're born with. You get them from your parental haploid genomes. They follow you throughout life. They certainly are related to many illnesses. There have been a number of genome-wide association studies looking at how germline variants influence everything from eye color and height to whether or not you might even get addicted to cigarettes if you try them. But those are very distinct from somatic variants, and somatic variants are what I'm going to tell you about today, particularly the role of somatic variants in this aging-associated disease. Somatic variants are the variants that we acquire. They're the reasons that we assemble or are assembled as mosaics. And you can see probably some evidence of this if you look at your skin. You've got areas where you've got marks that are a little bit different. They're sitting in islands. What's up with that? It's probably the somatic variation process playing out kind of over your body. And so these somatic variants do correlate with age. So this is just a couple of examples that I pulled from the literature. You can see that there's not a super robust correlation, although this is only 16 or so patients, but it is statistically significant. Likewise, if you look at exposures like tobacco use, and you look at current smokers, and you look at the somatic variants that are within lung tissue or airway tissue, you can see that there's a trend towards kind of an increase in the burden of those somatic variants in individuals who are smokers, and all of these things still correlate with age. So what we've done in our lab, and here I'm speaking about kind of what James DeGregory has done, is we took a look at all of the sort of published, genome-wide assessments of this somatic variation. And we kind of did a meta-analysis and looked across a variety of different tissues, how many of these variants are cropping up. And in particular, James is interested in understanding cancer and the origins of cancer. And if we just filter all those variants on the basis of the ones that we know are associated with cancer, like EGFR mutations or KRAS mutations, we find that almost across the board, in every tissue that we've looked, the number of cells harboring those mutations goes up with age. So these cells are developing these cancer-causing mutations. They are growing and outcompeting their surrounding tissue, and that the expansion of these cells has a tendency to correlate with just the time that we've spent on planet Earth. So that in and of itself probably raises a lot of questions about kind of the origins of cancer and what causes some of these cells harboring these bad mutations to break out of their environment and cause tumors. And that's something that the lab actively studies. On the other hand, or maybe in addition to that, we see that there are patterns of some of these mutations in individuals who have been long-term smokers. So in particular, the cancer-causing mutations have a tendency to be increased proportionately in individuals who have smoked for a long period of time. And I'll just highlight KRAS for you here, where you can see that folks who've never smoked have, a 3 to 5-fold less likelihood of harboring these typically cancer-associated KRAS mutations than individuals who smoked. Is that biologically significant? I can't tell you for sure. It's a relatively low fold number. We're talking about, a couple of cells out of several thousand that are harboring these mutations. But it's interesting, nevertheless, that when you aggregate that over the total number of cells that you have in your lung, you're probably harboring millions of these mutations by the time you've hit age 50. So, what are the consequences? Probably some impairments in how cells repair, probably some impairments in how cells deal with misfolded proteins, probably some changes to how cells signal that they're damaged. Maybe some of these cells are putting out chronic inflammatory signals that are, related to senescence, that may be related to those aging hallmarks that we talked about earlier. But the short answer is we don't really know yet. So, getting back to these learning objectives, somatic variation may relate to diseases such as cancer. Aging lungs accumulate these mutations. Certainly, we can see those clones growing and expanding and outcompeting their normal peers with the more time that they have to sort of cultivate. That certainly could be related to cancer. Is it related to other diseases? So to get at this question, we use a very sensitive DNA sequencing technology. It's called duplex sequencing. And the reason that we have to get really sort of in the weeds about this is duplex sequencing has the ability to pick up extremely rare mutations from a very error-prone system. So most of the DNA sequencing that we get, has a requirement to expand the DNA that you have so you can load it onto the sequencer. And all of that requires polymerases that are very likely to introduce mutations across the genome. So the way that we get around that is we essentially take our tissue, we take the DNA out of it, and we put barcodes, literally, DNA sequences that we can map back on either sides of those two DNA strands. So that when we're taking a look at all the data, we can look for that sequence of A's, C's, G's, and T's, and we can basically say, okay, this particular DNA fragment was paired with that particular DNA fragment. And if they both have basically the same mutation in them on both sides that have to be complementary by definition, then we can have a pretty high confidence that those mutations were in the original DNA strand. They weren't mutations that we introduced because of the way that we process the tissue. So we have a panel of about 31 cancer-related genes. I'm listing them for you here. So we're not looking at the whole genome because we have to sequence this so deep that essentially it boxes out how much ability we have to sequence genes that we aren't looking for, and that's a flaw in the research. So that's something to keep in the back of your mind. It's not a flaw; it's a feature, but it is. I think a limitation of this approach. But nevertheless, we can get some idea of what's kind of happening in these tissues. And so we're going to take lung tissues from David Schwartz's lab. We're looking at IPS and controls. This is the first 8 or so specimens that I've had a chance to sort of look at and analyze so far. So this is the research in progress part of the talk. We've actually identified 9 control specimens and 26 IPF specimens. So by the time this research is done at the end of this year, we'll have more numbers, not huge numbers, not genome-wide association study type numbers, but certainly enough to hopefully draw some inferences about whether or not we're seeing mutants that are in IPF that are not in controls, or whether we're seeing any patterns of mutation in IPF versus controls that might be relevant for understanding the aging related processes to this disease. And so we get a lot of information out of these data. What this sort of figure is going to tell us is the relative number of types of mutations that we have. So missense, meaning that the mutation causes a change in the way that the protein is coded. Nonsense, meaning that the mutation essentially causes a stop codon and interrupts the synthesis of one of those key sort of cancer-causing proteins. Frame shifts sort of have the same effect. Splice site mutations, basically how those genes are sort of constructed and then translated, and gives us an idea of kind of the relative proportions of different types of mutations. So T's to G's, T's to A's, T's to C's. This is important because these types of changes and the context that they occur in actually have some patterns that have been associated with different processes that I'll tell you about in a second. But then this also gives you kind of an idea that across these specimens, what's the percentage of P53 mutations that are occurring that are missense or nonsense or what have you. And just to kind of organize this a little bit, when we look at these mutation frequencies, the ones that are, basically occurring 100% of the time, even though they're sort of variants and they're not kind of specific for the 1,000 genomes sort of cataloged, aggregate ideal human genome. Those variants are probably germline, right? They're happening in 100% of the cells, so they happen very early kind of in division, and they're represented everywhere, right? Variants that are occurring at a kind of a variant allele frequency of about half, those are probably heterozygous. So again, probably germline. And then the somatic variants that we're, kind of interested in are kind of happening at this very low level. But there's a lot of them. And so even though the appearance of one specific variant in one specific gene might be on the order of 1 cell in 1,000, many of these cells are harboring more than one mutation, and many of them are harboring multiple mutations in multiple genes. So when we look at that and we try to identify sort of if there's a relationship between the types of mutations we're seeing and the genes that we're seeing them in, we can kind of look. Most of these boxes are black, meaning that the genes that we've sequenced, kind of listed for you over here, carry multiple mutations along the length of their coding regions that may be splice site, missense, in-frame, or others, but effectively these genes are just getting kind of rained down on in terms of the number of polymorphisms that they accumulate over the lifespan. And when we look, we can sort of see that there are different patterns to where these mutations are occurring in the genes. And so if we look, for example, at this gene FAT1, which is an acetyltransferase or a protocadherin that's sort of important for organ growth and kind of the YAP-TAZ pathway, we can see that there are focal points for where a lot of these mutations are kind of organized. Same thing with this TIAM2, which is a guanine exchange factor that's important for cell shape and cell signaling. On the other hand, if we look at Notch1, which is a developmental factor, we look at P53, we see more of a mixed kind of pattern, right? So Notch1 is just getting kind of carpet bombed. Everybody's got a mutation in Notch1 somewhere, but it's happening at a bunch of different locations. P53 has some mutations that seem to be kind of reproducibly occurring. And P53, of course, is the genome sentinel gene. It's the one that tells your body when you have, or tells your cells when there's a mutation that needs to be repaired and what to do about it. But also there are areas where it's just getting absolutely mutated all over the place. So even though the frequency of any one particular gene might be relatively low, there are a lot of cells that are harboring mutations in these genes at some location. And when we look and we compare those two controls to those 6 IPF specimens in this pilot cohort, we do see that there's an increase in somatic variants in pulmonary fibrosis. So if we ask the question, which of these genes are sort of differentially varied, and this is of course very simplistic and very early days, but we do see some genes that come out of this. So SMAD4 is important for signaling in the TGF-beta pathway. TGF-beta is thought to be important in pulmonary fibrosis. If anything, we would expect the signal maybe to go in the other direction. So maybe it's interesting, maybe it's spurious. But the point is that we can start to get a sense of maybe what the landscape is for these mutations in this aging-related disease. And I think that's where potentially this could be interesting. So getting back to this question about how mutations occur and which ones we accumulate. Every mutation has sort of a bias to it. The chemistry of the bases in your DNA, A, C, T, and G, set up in an environment where certain types of mutation may occur with greater or lesser frequency. And so if we go back to kind of this IPF data that I'm showing you, you can see that the number of cytosine to thymidine mutations is pretty high. The number of thymidine to cytosine mutations is likewise also kind of elevated relative to the number of Cs that are turning into As or Cs that are turning into Gs kind of throughout. And we divide these into transitions and transversions. A transition is just C to T or T to C. So kind of those small sort of ring-like nucleotides changing into the other ring. Or C to A, where we've got a small ring changing into a bigger ring. And those, for various reasons are less frequent, but they still happen. And you can see that both of these processes are happening at the same time, but that most of the samples kind of follow this pattern of those small transitions kind of occurring, Cs to Ts and Ts to Cs. And when we look at cancer genomes, there are actually ways that we can look at base signatures. And these are usually 3 nucleotides. And if you think about this kind of mathematically, there are 96 potential combinations of 3 nucleotides where you could have that middle nucleotide go from one base to a different base. And we can take this data and actually analyze it for these 3 nucleotide patterns, and we can come up with signatures that define specific pathologic processes. And so methylation becomes interesting here. So throughout the genome, if you methylate a cytosine, which usually happens on CpG repeats, you lose that methyl group Now it's a uridine, and a uridine is an RNA molecule, but it's basically synonymous with thymidine. So when your base excision repair mechanism goes in to change that base, it changes that C to a T. And that happens pretty frequently. The other thing that happens is you've got guanine in your cells. Guanine can get oxidized really easily by reactive oxygen species and by different types of oxidative inflammatory bursts. When that gets mutated, it oftentimes acquires an oxygen. Now it actually looks like an adenosine, and it'll pair with a thymidine when it gets repaired. And so that takes a C on the other strand and turns it into, I'm sorry. Yes, so the guanine oxygen actually will bind with adenosine. So that takes a C on the other strand and turns it into an adenosine. So anyway, so bear with me. So the point is that all of these different signatures have a way of sort of tracking back what it is exactly that caused the mutation to happen with varying degrees of success. Because you can imagine deconvoluting all of these signatures can be pretty computationally intensive. And of course, tobacco smoking is right up there. So does tobacco smoking leave a scar? Yes. It causes a lot of these oxyguanine changes where we're seeing cytosines turned into adenosines in the genome. And so when I look at the data, what I've found is that most of these signatures in the IPF specimens that we've looked at, although this also contains the control specimens, correlate with this somatic base substitution score number 5. Now, SBS5 doesn't really have a known etiology, but people have described it as being clock-like, meaning the number of these mutations has a tendency to go up with aging. And the etiology of it probably has something to do with that base excision repair pathway, but it's another interesting area potentially for future investigation. And so the conclusions are, we're all mosaics. Aging is associated with this progressive clonal mosaicism throughout tissues in the body. Many of these variants actually occur in cancer-causing genes and in genes that give the cells that harbor the variants an advantage over the cells around them. So we're thinking about EGFR, KRAS, things like that. Tobacco smoke increases the frequency of these mutations. It increases the burden of these mutant clones. And that probably occurs both because tobacco impacts the guanosine residues and causes more mutants, but also because some of the cells that are in there are probably getting used to having a lot of tobacco smoke around, and that gives them an advantage. And then we see some suggestion that in both cancer and non-cancer conditions, this process could be relevant to disease in older adults. And so IPF lungs do seem to be at least characterized by a higher burden of somatic mutations, but more work I think really is needed to sort of uncover that. And then somatic mutation signatures might give us some insight into pathophysiologic processes. In this case, we have an etiology that's unknown, but maybe that's a place for future research. Okay, so to follow up with this case report. So what are you going to do? I think as a primary care geriatrician, you've got this patient, they've now got a new cough, they've got their lungs full of kind of honeycomb cysts and fibrosis. I think you're probably going to want to get them oxygen, and you're probably going to want to see whether or not they're a candidate for one of these medications that can potentially preserve health span and preserve quality of life, and to keep that disease from progressing, particularly since this individual has loved ones that he needs to be around for. I think if this were an 84-year-old who had rheumatoid arthritis, we might actually implicate the rheumatoid arthritis. And so we might want to talk to our rheumatology colleagues about whether or not his immunomodulation therapy is the right one. There are other things that can cause somewhat similar patterns. One of them is hypersensitivity pneumonitis. That's usually due to aeroallergens that the body responds to, generates granulomatous inflammation. But when it burns out, it leaves these scars and these honeycomb cysts. And so you might want to make sure that, there's not mold in the home. You might want to make sure that the patient doesn't have like a hot tub. You might want to make sure that that pet bird that they've been taking care of for 70 years goes to one of their kids, or maybe to, a shelter. And so, and you might also think about sort of like drug and toxin exposures. So a lot of these folks that we're seeing, are, spent 40 years as roofers, or they spent 40 years crawling around attics. And so some of them are going to have asbestos exposure. Asbestos can definitely be part of this process. Doesn't change the therapy, but at least gives you some peace of mind that you've identified potentially a cause and that those exposures can be mitigated. Antifibrotics, there are really two. One of them is pirfenidone. Pirfenidone has been around for a long time. You take it 3 times a day. It leaves you with a horrifying sun-dependent skin rash. I don't recommend it particularly for patients who are gardeners. Nintedanib, on the other hand, is a sort of newer tyrosine kinase inhibitor. It actually works for various flavors of pulmonary fibrosis. So rheumatoid arthritis and hypersensitivity pneumonitis can be treated with nintedanib in addition to idiopathic pulmonary fibrosis. I know this is alphabet soup, but the point is that it's a twice-a-day drug. It's much better tolerated, does cause some GI upset, might be associated with some weight loss. But that's what most of our patients tend to be getting at this point. You probably should see about some pulmonary rehabilitation, particularly if your patient is very sedentary. And then you typically want to monitor these guys every 6 months or so as a way of sort of giving them both advanced care planning and also as a way of sort of deciding whether or not the therapy that you picked is going to work out for them. Okay, so germline and somatic genetic variation. Germline you get from your folks, somatic you acquire. Somatic variation may be related to certain diseases such as cancer. I think the court's still out about pulmonary fibrosis, but I think there's an intriguing signal that maybe more of these mutants are kind of aggregating in individuals who do have IPF. And I think that that's an interesting, potentially question for basic research. And then general mechanisms that can cause these mutations to occur. I think the principal one is smoking. Smoking mutates your cells. It selects for aberrant cell clones in particular that have mutations in cancer-causing genes like KRAS. That's probably what's happening to folks who develop lung cancer. Don't smoke. End of rant. All right, so none of this would be possible without everybody kind of in the lab, who's helped with these projects. Particularly, I want to shout out Ed Evans and James DeGregory. And then I want to give a shout out to Ivana Yang and David Schwartz, who collected all these IPF specimens that we're able to use. They're great collaborators. The University of Colorado Cancer Center, Bioinformatics and Biostatistics Shared Resource and Genomics Core did all the analysis that gave us the variants to start to call. And then the GREC funds and protects my time, and the CCTSI funds the research that I'm doing. They're exceptional resources if you're a junior faculty member and you're trying to figure out how to move your research forward. I would look very closely at both of them.
Thanks, Jonathan. That was a fantastic presentation, and I learned a lot about how aging impacts pulmonary fibrosis and potentially cancer. So I appreciate the basic science research there that goes in, and the way you rolled it into a clinical case. That was really nice. Thank you. Questions from anybody?
Thanks for the talk. I have a theory that I made up, so it might be totally wrong, about IPF, that it seems to me like the clinical course of people who are diagnosed in like their 80s and 90s versus younger people looks a little bit different in terms of like quickness of progression and flares and other things. But that might not be true. But I guess that makes me wonder, is the genetics of people who get ILD or IPF when they're younger different than older? And do you think that maybe those are kind of different diseases or the same? And if I'm way off base, you can tell me.
Yeah, no, I don't think you're off base at all. It's a great question. So hopefully everybody online heard it. But in essence, I think that there's a lot that we're learning about kind of the natural history of pulmonary fibrosis that probably bucks a little bit of what I've told you kind of like here today. So what I would say first is that, number one, we are definitely ascertaining, finding a lot more pulmonary fibrosis, because we know it's out there. People are looking for it. We're getting two-dimensional imaging on everybody every time they come into the emergency room. And so we're seeing like a lot of these sort of features of pulmonary fibrosis, which may or may not have a lot to do with kind of the classic sort of picture. And so we see kind of, these areas of reticulation, reticular change, maybe not honeycomb cysts in individuals, and then they wind up coming back to their primary care doc and not knowing what to do with that information, because up to that point they were asymptomatic. I think the point is that you're right on. There probably are a lot of different flavors of pulmonary fibrosis. We know for sure that there are some variants that cause IPF very early in pediatric populations, right? I didn't talk about those polymorphisms, but if you have a mutation to surfactant protein C, certain types of mutations, then your alveolar type 2 cells die and you get IPF, you get pulmonary fibrosis in childhood. We don't see those obviously in adults quite as much. So a lot of these other genes probably are filling roles that have different functions and have different latencies to them. The telomere story, I think, is really interesting, and I'm happy to talk to you about that also kind of offline, because a few other folks have their hands up. But to get to the basis of your question, yeah, I think there are a lot of different endotypes. I think that when disease became severe enough for us to notice in the past, when that life expectancy data was probably analyzed, that was before we had the advent of these antifibrotic therapies. We might have only been looking at the most severe cases under those circumstances. And what divides a case from one that rapidly progresses or progresses at all, between one that probably is there but is relatively stable over time, we don't have a good fixture on.
Thank you so much. My question was, what is the role of immunosenescence and like the changes that we see within the immune system in this picture as well? And if that's like outside the scope of this, that's totally fine.
I'm more than happy to talk about that. I think it's really interesting. And the short answer is we don't know a whole lot yet. And I think the overall sort of question about the immune system and how it changes over time, and whether or not a lot of the manifestations of aging that we see are really due to a malfunctioning immune system, is like a really fundamental question. So, we do know that a lot of these variants that are in clonal hematopoiesis have a tendency to push the hematopoietic system from an adaptive immune sort of context, where we're getting lymphocytes that can provide a little bit more targeted sort of therapy to address pathogens or bad acting cells, to much more of an inflammatory kind of macrophage, neutrophil, granulocyte sort of phenotype, right? And there are lots of signals in pulmonary fibrosis tissue that suggest that those types of inflammatory pathways, IL-1, IL-6, TNF alpha, probably play a role. But we haven't done the population studies yet to be able to say, whether or not we see these phenotypes in individuals who, for example, have clonal hematopoiesis. You would expect that would be like really low-hanging fruit, and that the NHLBI should be all over that. And it's possible people have worked on it and didn't get a result and then didn't publish it. But we haven't seen a whole lot of data along those lines yet. So I think your suspicions that immunosenescence probably plays a role in the lung is probably perceptive. I just don't know that we have any real definitive data to nail down. I will say that one of the advantages of some of the emerging techniques to get these duplex sequencing signatures is that we're getting better all the time at essentially increasing the specificity of those variants that we find, and the system and the technique itself is getting better. So we are moving out to more of a genome-wide sort of somatic variant analysis. In fact, I just submitted a CCTSI proposal about that, if anybody's involved in that. But I think, and I think that could be one opportunity for us to pick up some of these the clonal hematopoiesis in the lung, but so far there hasn't really been any definitive studies along those lines.
Hey, thanks for the presentation. I've got a comment and a question. I just appreciate that update on median survival in IPF. I feel like when I was taught, median survival was 2 years, but totally the lag time to diagnosis is way shorter now because we image everybody. And so that's nice to know just as far as counseling is concerned and clinical practice. The next piece of the question was just, I know you'd mentioned that the sort of omic profile of gene activation and the samples that you collected were age-related or related to that SBS5 clock-like pattern. I also am aware that IPF is really common in veterans and thought to be due to an Agent Orange exposure. So I was just curious where the samples were coming from, because if it's exposure-related, then I would also imagine then the profile should be different than SBS5.
Yeah, I think that's a really interesting question. So I would say that one of my favorite stories about these SBS signatures, and you may be familiar with it, is a compound called aristocolic acid. So aristocolic acid has its own base substitution signature. Aristocolic acid is actually a toxin that's produced by plants that for many years apparently was causing renal fibrosis in individuals in the Balkans. And the etiologic agent of this was not at all clear. It was clear that everybody who developed this renal fibrosis had this pattern of mutations, but it wasn't until people did some very, very cool epidemiologic work to understand kind of like what the actual sort of source and commonality between those individuals was, that people began to understand that actually there was this weed that was getting picked up when people were harvesting wheat, and that weed seeds were going into the flour that was being made for the bread that these people were eating, that was actually mutating their DNA and causing some of this stuff. And there are other examples of that, like ecologically. So aflatoxin, which is produced by Aspergillus, also causes some of these sort of similar mutation patterns. Nobody's looked, as far as I know, and I'm certainly not an authority on the topic, at specific mutations related to Agent Orange. People certainly looked at dioxin-related mutations. And so I think the question of whether or not there could be sort of a signature for that in veterans who are exposed to Agent Orange is a really interesting question. As far as the samples themselves, so some of the samples came from this great biorepository at NHLBI called the Lung Tissue Research Consortium Biorepository. And this was, I think, started in 2006. It has clinical demographic radiologic data, pulmonary physiology data from, I think, 3,000 to 6,000 individuals at this point. And there's varying overlap with what data you can get from which individual, et cetera. But that's all sort of out of the NHLBI. So 3 of the specimens came from that cohort. So theoretically, the individuals who were in that cohort who got lung biopsies would have been, you know, kind of in their 60s at around that time. A little bit early probably for Agent Orange, but not out of the realm of possibility. But a lot of the other sort of specimens have been a little bit later. I think, you know, but I think the question is a good one. And it's definitely something that I'm very aware of. I submitted a proposal to the Military Exposures Research Program. It got triaged, but it's definitely something that's on my radar as far as like following up, because I think if we could identify some of these signatures in individuals who, for example, have deployment-associated lung disease, that would do 2 things that would be really helpful. One of them is we might be able to kind of get an insight as to whether or not they're accumulating these cancer-causing mutations, and maybe give the VA some advanced warning about that as a possibility. So we could look forward in time potentially. The other thing is we could look backward in time and maybe try to identify whether or not there are some of these sort of stereotyped mutation patterns that are worth looking into. But so far, nobody, as far as I know, is doing that.
Hey, thanks for a terrific talk. Building off of Elizabeth's question, my question was around senolytic therapy, so senescence targeted therapy. To my knowledge, there's been 2 published trials using dasatinib plus quercetin as a senolytic therapy for patients with idiopathic pulmonary fibrosis. And just from those small trials, it appears to be at least safe, tolerable, and potentially efficacious.
Yeah.
Just curious to get your thoughts on where that might be headed.
Yeah, so Jim Kirkland is sort of the person who's been pushing those studies up out of Mayo Clinic, and it's been really kind of an amazing story to sort of watch it evolve from like 2017 in the mouse models to, you know, sort of like human trials like 2 or 3 years later. What I can tell you is that it seems to be stalled in phase 2. I don't know why. I suspect there may be an efficacy endpoint that they're lacking. It may be that they might need to be recruiting a lot more patients. I'm not really sure. He actually gave a talk at a conference in China that James DeGregory attended a couple of weeks ago. He really didn't talk very much at all about dasatinib quercetin in the context of pulmonary fibrosis. So it seems to be fizzling. I'm not sure what happened. Yeah.
Thanks. We are out of time. Unless there's anything online? Nope. All right.
Thanks for attending, guys.